So I just wanted to give a quick introduction about myself as well. So I'm a research faculty at Yale using AI to translate geroscience to clinic. Previously, I did a PhD under Dr. Morgan Levine, who's currently the VP of Computation at Altos. I previously built a computational biology startup called Elucidata that's now evaluated at upwards of $120 million. I patented a biomarker, which Andrea just told you about, called SYMPHONYAge, which is licensed to True Diagnostics, and I'm a director of data science at Healthy Longevity Clinic, and I'm a principal at a venture capital fund called Longevity Tech Fund, and angel investor in a couple of companies, which you might see on the right-hand side. I'm not sure if that's kind of visible to all of you guys or not. But let me start with some very basic things here. Geroscience or longevity science is making headlines. I'm not sure if you might have heard about this really cool drug, but it's called Ozempic. It's making everyone lose weight, but it's a really cool drug. Even Serena Williams is taking it, apparently. But it's supposedly also a gerotherapeutic, or people claim it can extend lifespan. I don't know if it does, but some people claim it does. And the Wall Street Journal even reported about it. The Kardashians, I don't know how many of you know who the Kardashians are, but Kim Kardashian took the biological age test by True Diagnostic. Check out season 4 episode finale, or I think the season finale of season 4 to check out which one of the Kardashians has the lowest biological age. And this guy, how many of you know this guy? Brian Johnson, right? He spends like everybody knows Brian Johnson. He spends like $2 million a year to stay young forever, right? So clearly, people across the globe are interested in longevity. They want to stay young forever. And people might think it's a rich people thing. And the reason for that is because translating science from these headlines to patient might seem quite easy. So think about it this way. How an average Twitter, or well, now X user might see geroscience or longevity science as. So think about a hypothetical person called Brian Bonson, just for legal reasons, not any other reason. So Brian Bonson goes to a longevity clinic. He measures a couple of their clinical biomarkers to quantify their aging. Based on these biomarkers, the results, the clinician suggests a couple of interventions. A few months later, the clinician assesses JB's biomarkers to see the intervention's impacts, and JB leaves the clinic feeling like an 18-year-old, right? That's how we would like the longevity clinics to be. If this was the case, all of us would be practicing preventative longevity medicine, right? But that's not the case, right? That is not how longevity medicine is right now. There are issues at each of these steps. So for example, at the very first step, there's the issue about measuring sort of biomarkers to quantify aging. We do not have sort of these insightful aging biomarkers that can really sort of quantify aging in an insightful way, that can tell clinicians how is sort of aging really being affected, and I'll tell you what we are doing to improve that. The second is, we don't really know which interventions are actually truly geroprotective, right? And which intervention might work for me might not actually work for you. And the third thing is, which biomarkers are actually responsive to interventions, right? We don't quite know that either, right? And which of these are reliable biomarkers, right? Which of these fluctuate during the same day, right? At an n equal to 1 level, these biomarkers work at a population level, but do they work at an individual level? We don't really know that. So we have to solve these challenges before we take all of these things to clinic. So let's talk about that. So which of these how do we make these biomarkers insightful? So to start with, what exactly is an aging biomarker? So if you think about an aging biomarker, at the very simplistic level, it's some kind of a biological data that's predicting some kind of a health outcome, right? So it could be some phenomic data like your MRI, it could be a clinical biomarker, it could be a proteomic data. I really like DNA methylation data because that's what I did my PhD in, so I'm biased towards it, but it could be any other readout, right? And it's predicting some kind of sort of output, like it could be time to death, like how many years are left in your life. Sounds a little gloomy. It could be how many years have been spent, or the number of candles on your birthday, or how much time is going through that particular sort of hourglass, right? The pace of your aging. The one that I like is DNA methylation, which essentially tells you, is a measure of how much of a particular gene in your body is methylated or unmethylated, right? So what that means is that every gene, or as you might know in your DNA, right, has this kind of group, this sort of group called a methyl group that essentially allows for more expression or less expression of that gene or not. At a very simple level, think about this as a recipe for life, right? And I'm simplifying this a bit. So if your DNA is the perfect sort of recipe itself, this recipe over time sort of fades away, right? This recipe sort of fades away. This DNA methylation over time fades away. And we can quantify how much of this recipe has faded away over a period of time, right, using a machine learning algorithm, right? And that is effectively what this aging biomarker does, right? We're quantifying how much of this recipe has faded away. So just like this recipe that I have that has a lot of fading, a lot of blotches in it, what we are doing is quantifying how many blotches are there in our aging biomarker, right? And that is effectively what our aging biomarker is. It's telling us how old is our recipe. And these epigenetic clocks or these epigenetic biomarkers have been shown to be very prognostic, what means that they have been predictive of mortality, of cancer, of cardiovascular disease, so on and so forth. But the issue with them is that they are pretty much very one-dimensional. They don't quite predict anything more than telling you that you're 5 years older or 3 years younger. Now, that's not very insightful for a clinician. What will a clinician do with the fact that you're 5 years older or 3 years younger? A clinician can't do much about that. They'll just be like, OK, you're 5 years older. What do I do with that, right? A clinician sees your body as a physiology of different organs. That's how a clinician has always studied sort of their biology of things. That's how a clinician, sort of an MD, essentially goes through their education, right? And so that's why we built sort of this thing called systems age, which is an organ-specific measurement of clocks from a single blood draw, which is about 1 billion data points from a cohort built around some AI models. And we predicted these 11 different organ-specific clocks. This was built along with Dr. Morgan Levine, who's now at Altos, was previously at Yale. And we combined it into a whole body clock. And you can read more about this in Nature Aging. But to summarize this, we have validated this in 8 different cohorts and 20K plus subjects. And as you can see, for example, for cognitive function, the brain score is really predictive. The musculoskeletal score is really predictive of physical function, so on and so forth. For coronary heart disease, the heart score, for arthritis, musculoskeletal score, so on and so forth. But what this test really does is tell you that if you're 5 years older, now you can really know the reason you're 5 years older is because your heart score is 11 years older than it's supposed to be, right? Or your lung score is 7 years older than you're supposed to be. But your kidney score is a lot younger, right? So you don't need to focus on your kidneys. You're fine with your kidneys, right? And so your clinician can actually target those particular organs with specific interventions, and I'll tell you how we can do that. And this tool is now available through as SYMPHONYAge by True Diagnostics. Talking about effective geroprotective interventions, one of the biggest challenges is which interventions actually work best for which groups. And so what we did was we actually collated 50 different clinical trials across 110 different biomarkers, right? So there's never been this kind of analysis that has ever been done. And we looked at 15 commonly used biomarkers in the space, and we did a very simple analysis. We sort of collated all of this data together for these 15 commonly biomarkers, and every little dot that you see is the effect size for one whole body clock. And what we saw was there were a few interventions that essentially decrease biological age very effectively, and others that increase biological age, but most of them decreased it. So for example, something like metformin, a very common diabetes medication, decreased biological age. In general, all longevity interventions of the 51 decrease biological age. Most effectively, lifestyle and pharmacological interventions were the ones that most effectively decreased biological age. What we also saw was a very specific effect with certain interventions. So for example, intramuscular plasma concentrate, which was a type of plasmapheresis done specifically muscularly, had a very specific effect on the musculoskeletal level. Metformin, for example, had a very multi-system effect, while something like hyperbaric oxygen therapy specifically had a very lung-specific effect. And again, I can show this for every specific organ and every specific of the 50 interventions that we did. But the really powerful thing is we can now essentially map a particular intervention to a particular increased aging pattern. So if a particular individual has increased aging in a particular organ, we can tell them which particular interventions to take based on our particular increased system aging patterns. So potential to match interventions using system specificity. And the last point is responsive and reliable aging biomarkers. And so what's really interesting is, again, we can go back to this map and invert the analysis and say, which of these biomarkers are actually responsive across the board for these 15 interventions? So each little dot that you see is the effect size of one intervention. And what we see is that only a handful of the 15 biomarkers that we have are actually responsive across the board. Again, I'm going very quickly over this data, but this is very significant data. What this shows is that only these second-generation biomarkers that are really very rate of aging mortality clocks are the ones that are really predictive and responsive to these interventions, which are across the board. But what's really exciting is that not all of these clocks that we have are actually reliable. What that means is that they change quite dramatically during the day. So if you measure them in the morning, they'll tell you you're 5 years younger. If you measure them in the evening, they'll tell you you're 6 years older. And we did an analysis where we look at technical and biological ICCs for these clocks. And we saw that most of them fall in this moderate to poor ICC. But we were actually able to improve them quite dramatically and move them to the excellent ICC region by doing some improved machine learning. And we saw by doing that, you actually require significantly less samples in actual clinical trials by decreasing this and improving the signal-to-noise ratio here. So in practice, when we do all of these things, we can actually use these clocks at an individual level in patients. So I'm actually showing this data from our longevity clinic in Prague, where this individual had accelerated aging in 4 systems, in the musculoskeletal, lung, metabolic, and immune system. They had a chronological age of 51 years, history of autoimmune disorders such as arthritis. They had a BMI of 31, and they were a current smoker. They were given 3 interventions, 4 actually, but we had data for 3 of them. But lung aging, so smoking cessation, which would decrease their lung aging, intermittent fasting, and metformin. Nothing too crazy, right? So pretty normal interventions, right? We knew that smoking cessation would target their lung aging, intermittent fasting, metabolic and musculoskeletal aging, and metformin will have a multi-system effect. What we saw after 1 year was they had strong medication adherence to IMF, metformin, and NMN. They continued smoking cigars, but decreased smoking cigarettes. They lost about 9.5 kgs and had qualitatively less arthritic pain in the last 3 months before their blood draw. And that could be seen very clearly in their DNA methylation markers as well. So their immune age, as measured by their DNA methylation SYMPHONYAge, went down significantly. Their metabolic age went down significantly. Their musculoskeletal age went down significantly. Their lung age did not change because they continued smoking cigars. And you see a bunch of their other organ scores also went down because of the multi-systemic effect of metformin, or at least that's what we hypothesize. So, but the really amazing thing is you can actually go to this website called translate.io and replicate all these results and do this analysis for yourself. We have all of this data available for you. So in conclusion, what I'd like to say is that there are many challenges that exist in translating the science to clinic, and there are solutions in progress that we are working upon, and we're working to bring these solutions to patients. And I'd like to thank you for attending this talk. Thank you so much.